Hello and welcome to Group 14's Advanced Cell Biology Multimedia presentation, titled Exocytosis versus Endocytosis. How do they differ? Both exocytosis and endocytosis are key processes for the transport of molecules that cannot passively permeate the cellular membrane, so how do their pathways differ? Both processes utilise vesicles and their interaction with the plasma membrane in the transport and storage of material. Endocytosis facilitates the movement and use of molecules into the cell via vesicle formation, whilst exocytosis facilitates their movement out alongside eventual vesicle destruction. Let's take a closer look at each pathway in detail, both in the nature of the material they transport and the mechanisms in which they utilise vesicles to achieve their function. Now for a closer look at the exocytic pathway.
The exocytic pathway serves to direct secretory vesicle contents to the plasma membrane, be it to release waste materials or extracellular matrix components out into the extracellular space, or to deliver lipids and membrane proteins for incorporation into the membrane itself, expending energy in the process. There are two types of exocytosis. Constitutive exocytosis is unregulated and performed in every cell. This type aids toward the functions described earlier of either waste removal or membrane component fusion, post-transport vesicle fusion also. This type therefore constantly occurs, as both functions are continually required for proper cell function. Regulated exocytosis is mediated, requiring specific stimulation, intracellular calcium, and sorting signals before fusion and contents discharge can occur. This regulation is necessary and pivotal in the release of specific hormones and neurotransmitters, for instance, unmediated release of such substances potentially leading to serious pathological states. There are 5 general steps to achieve this. Number 1. Vesicle trafficking. Proteins that are to be excreted begin their journey at the endoplasmic reticulum. From there, they are translocated across the endoplasmic reticulum and into the lumen. The next step involves them being selectively removed and packaged into vesicles before being transported to the Golgi complex. These proteins then travel through the trans-Golgi network, where they are modified to fit their specific functions. This whole process is directed along the microtubule network. Number 2. Vesicle tethering. Tethering involves the initial loose interactions between a secretory vesicle and exocytotic sites on a target membrane. This occurs over distances of more than half the diameter of a vesicle from a given membrane surface, averaging greater than 25 nanometers. Mediated by fibrous rod-shaped tethering proteins, they are able to form a weak bridge between the two membranes, holding them in close proximity. The proteins that confer specificity between the tethering protein and transport vesicle are called Rab proteins. They are Ras-related GTP binding proteins that are responsible for binding selectivity. At this point, over 60 different types have been identified. Each compartment has a unique surface identity, which means each trafficking step in the exocytotic pathway requires a different Rab protein. In their GTP-bound state, Rab proteins recruit cytosolic tethering proteins to specific membrane surfaces. Certain Rab members are specific to each compartment membrane. For example, Rab5A is found on the plasma membrane. Rab2 is found on the Golgi. Rab proteins are GTPases, therefore needed GTP to activate them. Number 3. Vesicle docking. Docking is the process during which the vesicle and plasma membrane line up in a fusion-ready state. The vesicle is then able to dock and fuse with the plasma membrane with the help of snare complexes. Exocytosis requires additional specialized proteins that ensure vesicle docking is specific. The membranes of the vesicle and target compartment come into close contact due to interactions between the cytosolic regions of integral proteins. Proteins that secure these interactions are snares. They can be divided into 2 categories: V snares that are incorporated into the membrane of transport vesicles, and T snares that are located in the membranes of target compartments. As the two membranes get closer, the snare motifs of the T and V snare molecules from each membrane interact. Number 4. Vesicle priming. This stage includes all of the molecular rearrangements and ATP-dependent protein and lipid modifications that take place after the first docking stage but before the actual fusion occurs. Number 5. Vesicle fusion. The two pure regions of T and V snares then mix, causing vesicle membrane fusion, without concurrent fusion of the snare proteins themselves. The force produced from aforementioned T and V snare interaction is sufficient enough to cause fusion. Snare proteins are specific to the vesicle, enabling certain products to be moved to selected compartments, different combinations of V and T complexes being responsible for targeting different vesicle compartment interactions. Once fused, the vesicle remains docked at the membrane in a state of inactivation until receiving calcium-mediated activation signal discharging its content. While the lipid bilayers are fused, the snares from each membrane become part of the same membrane. The snare protein complex then dissociates due to a cytosolic protein NSF attaching to the snare protein and using energy from the hydrolysis of ATP to break the fusion. What happens to the vesicle? This kind of transient vesicle fusion is known as kiss and run. It is incomplete fusion between the secretory vesicles and the target membrane that causes the release of the intravesicular content. Full fusion exocytosis involves the complete merging of the two phospholipid bilayers in an aqueous environment.
And now a closer look into endocytosis. Endocytosis in the endocytic pathway is, in the broadest sense of the term, the reverse of exocytosis, in that it is used to bring components of the extracellular space into the cell. However, the mechanisms used bear distinct differences to each other. This process is used to internalise not only molecules that are too large for channels and simple diffusion, but also for receptor internalisation. There are two broad types of endocytosis, each with two subtypes in each. The first of which, bulk phase endocytosis, also known as pinocytosis, is a non-specific uptake of extracellular fluid alongside any type of molecule in it becoming trapped inside the vesicle to be internalised. This form of endocytosis also plays a role in the recycling of membrane portions from the cell surface between itself and interior membrane compartments. Macropinocytosis is a form of pinocytosis on a large scale, an energy-expending, actin-dependent process occurring in highly ruffled regions of the membrane. A pocket forms in which the cell membrane will invaginate, subsequently budding off into the cell. These are typically between 0.5 and 5 micrometers in diameter, and eventually form a large endocytic vesicle called a macropinosome. Similar, unselective uptake also occurs, though this time of large solid particles, in phagocytosis. This, however, only occurs in a select number of cells, such as macrophages and neutrophils, and is involved primarily in immune defense rather than in general cell function, recognizing pre-marked material such as bacteria and particles for internalization and destruction. Here is a quick schematic. Unbound phagocyte surface receptors will not trigger phagocytosis. Phagocytosis only triggers when the ligand-coated particle binds to the phagocyte surface receptors. Induction of particle uptake by the phagocyte occurs due to actin filament contraction, causing folds to invaginate a particle. The forces that contribute to deforming the plasma membrane to bring the material into the cytoplasm come from the polymerization of actin filaments.
The other main type is that of receptor-mediated endocytosis, the receptor-dependent uptake of specific extracellular molecules such as growth factors or hormones after they have bound to the receptors on the extracellular side of the plasma membrane. Said receptors concentrate in coated pits at very high levels in certain membrane domains. This type of endocytosis is both selective and efficient, even at low concentrations of said macromolecule. There are three main types of receptor-mediated endocytosis that can be observed. The main type is clathrin-mediated, the formation of clathrin-coated vesicles aiding towards the selective nature of this form of endocytosis at the cell membrane and in other vesicle trafficking pathways. This type of endocytosis can be broken down into the following steps. The first, binding of an adapter protein that is usually heterotetrameric, such as the adapter protein 2 complex, causes clathrin to be recruited via beta-adapted subunit action. These adapters remain between the clathrin lattice and vesicle surface, and each subunit confers certain functions. The Mu subunit, for instance, interacting with the specific plasma membrane receptor tails, causing accumulation of specific target molecules into the vesicle, again conferring selectivity. The second, the budding of the vesicle beginning to form, the clathrin lattice continues to grow, with polymerization of the clathrin inducing membrane curvature. Additionally, polymerization of actin filaments helps to overcome high turgor pressure, allowing further invagination of the coated pit. Scission of the clathrin-coated vesicles from the membrane is mediated by dynamin-related proteins. Subunits concentrated in the region assemble around the neck of the vesicle, forming a ring. GTP-mediated hydrolysis, induced conformational change, leads to separation of the budding vesicle from the plasma membrane. Finally, once the vesicle is pinched off from the membrane, it loses its clathrin coat, allowing for rapid recycling of the molecule for further lattice formation. The remaining two types of endocytosis are dependent on lipid rafts for internalisation. Specialised glycoprotein dense areas consisting of glycosphingolipids and specific protein receptors. They are thought to be affiliated with membrane protein and receptor trafficking outside of the classic receptor-mediated endocytic process. The first is calveolin-mediated. Caveoli are invaginations of the cell membrane that have been implicated in endocytosis of membrane components such as integrins and glycosphingolipids, as well as bacteria, bacterial toxins and viruses. This endocytic pathway requires unique signalling and structural machinery, indicating that it has distinct mechanism from other forms of clathrin-independent endocytosis. There are also several pathways independent of clathrin and caveolin that are much less common, often using cholesterol and glycosphingolipid-enriched membrane domains to influence internalisation.
The endocytic pathway. Once molecules have been internalised by the cell, they pass through a distinct endocytic pathway. Molecules are first delivered to the early endosomes. Here they undergo efficient sorting. For example, the high H+ concentration within allows for ligand dissociation from housekeeping receptors. Receptors involved in housekeeping roles, such as LDLs or transferrin, that have been internalised are recycled back to the plasma membrane via specialised tubular recycling compartments in early endosomes for continual housekeeping regulation. Receptors involved in signalling that require downregulation will be transported to the late endosomes and lysosomes for degradation. This process is often mediated by tagging receptor tails enzymatically with ubiquitin. Similarly, components of the extracellular matrix that have been taken in via bulk phase endocytosis and need to be broken down, such as phagocytosed bacteria, will be transported to late endosomes and lysosomes for degradation. There are also routes of transport between this endocytic pathway and the biosynthetic pathway for delivery of endocytosed molecules needed for metabolism. It is worth noting that this is a very complicated and interlinking pathway.
With all of this taken into account, we can evaluate the similarities and differences between endocytosis and exocytosis. Direction of movement.
Exocytosis carries molecules out of the cell, and endocytosis transports molecules into the cell.
Mediated trafficking.
Exocytosis uses RABs and snares, whereas endocytosis uses clathrin, caveolin, and lipid rafts.
Vesicle contents.
Exocytosis is used for carrying neurotransmitters, hormones, and cellular waste, whereas endocytosis is used for carrying nutrients and proteins.
Physical fate.
In exocytosis, the vesicle can be recycled, i.e. the kiss and run mechanism, or can undergo membrane fusion. In endocytosis, the vesicle travels to the endocytotic pathway, where it is then fused with early endosomes.
And energy used.
Exocytosis uses ATP and GTP, but endocytosis only uses ATP.
Thank you for listening.